last topic is how cells use genetic information to make proteins. I mentioned this earlier in some of the recordings about how RNA is used to make protein. The fundamental question is, how do proteins differ? On a basic level, proteins are different because their amino acid sequences are different. In other words, the order of the amino acids makes proteins different from each other. The central idea or central dogma is that the information on the DNA is used to make RNA. The RNA then is used to build a protein. The DNA contains instructions for putting amino acids in the correct order for a particular protein. Think of it like a recipe in a cookbook. The RNA is going to play a role in expressing this information. There are 3 types of RNA for this process: messenger RNA or mRNA, ribosomal RNA or rRNA, and transfer RNA or tRNA, which we talked about briefly in the last chapter. Let's look at this process. The DNA is in the nucleus of your cells. Within the nucleus, the information on the DNA is copied to make a message. This message is going to have the code or recipe for the protein. This process is known as transcription, and the product of transcription is mRNA. In this process, the information on the DNA is being transcribed or rewritten to produce the mRNA. The mRNA exits the nucleus and enters the cytoplasm of the cell. In the cytoplasm, the information is used to assemble the chain of amino acids, and this process is known as translation. Translation involves a ribosome, which is made of rRNA. The ribosome moves down the mRNA, and as it does, it assembles the amino acids into a protein chain. The information stored on our DNA is known as a gene. A gene is a large section on a segment of DNA that codes for a particular protein. The code is known as a triplet code. This means each amino acid in a protein is equal to 3 bases on the DNA. Let's look at this process of transcription. In transcription, the product is mRNA. The mRNA will be the message for the protein. In the first step, the DNA will unwind and separate. One strand will serve as the template strand. An enzyme called RNA polymerase binds the template strand and moves along it. As it does, it will add complementary base pairs. Now, because we're making RNA, uracil will be substituted for thymine. Once the RNA polymerase has produced its messenger RNA, the newly formed messenger RNA will exit and the DNA strands will unite. The information on the mRNA is in codons, so 3 bases on the messenger RNA stands for a single codon. This becomes important when we discuss the translation process. During translation, we will use the information on the messenger RNA to assemble amino acids in the correct order in order to make a protein. An anticodon resides on a tRNA. The anticodon has 3 bases and will be complementary to the bases in the codon of the messenger RNA. The start codon represents the start site for translation. First, a tRNA will bind to the start codon by complementary base pairing between the anticodon and the codon. The second tRNA with the second amino acid attaches will form a pairing between its anticodon and the complementary codon on the mRNA. Then the ribosome will shift down the messenger RNA toward the stop codon. As the ribosomal RNA moves, the first transfer RNA dissociates, but before doing so, its amino acid will form a peptide bond with the second amino acid. Now there's an open codon, and our third transfer RNA comes in with its anticodon and binds to the codon. This process adds amino acids to the chain until we reach the stop codon. Once the stop codon is reached, the polypeptide and everything else dissociates. The central dogma refers to the flow of information from DNA to RNA to protein. DNA has the information in the form of triplets. Each triplet is a group of consecutive bases that codes for a particular amino acid. This information is stored in the sequence of bases in DNA, so this is often referred to as the genetic code. Messenger RNA is formed by transcription and has codons that are complementary to the DNA triplets. Each codon corresponds to a particular amino acid during translation. At the ribosome, the tRNA anticodons deliver amino acids that are specified by the mRNA codons. The amino acids are linked by peptide bonds to form proteins. The correct order of amino acids is obtained due to the matching of the anticodon on the tRNA with the codon of the mRNA. You do not need to memorize these tables, just need to understand how to apply this information, which we will practice in class. The last page is a homework assignment which you should work to complete. This ends this chapter. You are encouraged to use the learning outcomes to create a study guide for this chapter.